Hello everyone, welcome to another warm-up. Let's get going. What are the three different mechanisms that cells employ to break down proteins? So there's the ubiquitin mechanism with ubiquitin protein ligase. Proteins can be broken down in the lysosomes, and then there are calcium-dependent enzymes as well. Next, what are the components of the blood-brain barrier? So there are three structures that you need to know. We have the astrocyte foot processes, we have the basement membrane, and then we have capillary endothelial cells with tight junctions, which are also called non-fenestrated endothelial cells. Next, a woman involved in an accident cannot turn her head to the left and has a right shoulder droop. What structure is damaged? So this is probably a right-sided sternocleidomastoid muscle that is responsible for the head turning. The right spinal accessory nerve innervates the muscles responsible for these actions. All right, guys, that brings us to the end of the warm-up. Let's get to that lecture.
In this video, we're going to discuss neurotransmission, specifically neurotransmission in the autonomic nervous system. We'll review how neurotransmitters like acetylcholine and norepinephrine are actually made and how they get released and what happens to them once they're released. So let's start with acetylcholine. In your study guide, you're going to find a diagram of a cholinergic nerve terminal, which we're going to label. Now, this gets a little complicated, and there's a lot of information here, but you do need to know this for your exam. This is about a 3-star topic. So, how does acetylcholine get released? Well, it starts off with the synthesis of acetylcholine. So here in the upper right, you have two things coming into the presynaptic cell. The first thing being choline. So choline is essential to making acetylcholine. And how does choline get into the cell? Well, there's a sodium cotransporter that brings both choline and sodium into the cell. So the substance that actually drives the transport of choline into the cell is sodium. And this cotransporter can be inhibited by a drug called hemicolinium. Hemicolinium inhibits the transport of choline. Once choline is in the cell, it gets bound to acetyl-CoA. And the enzyme that catalyzes this is called choline acetyltransferase. So choline acetyltransferase takes acetyl-CoA and choline and makes acetylcholine. And that acetylcholine gets packaged into a vesicle. Now, there's a drug that inhibits the packaging of acetylcholine into vesicles, and that drug is visamicol. So when visamicol is around, acetylcholine can't be packaged into the vesicle. So the presynaptic cell is storing acetylcholine in vesicles until it's needed. So what needs to happen for the acetylcholine to be released into the synapse? Well, the substance that's essential for this process is calcium. Calcium is very important in a lot of biological processes, in muscle contraction, in platelet function, in activating the clotting cascade. But here, the influx of calcium triggers exocytosis of these vesicles of acetylcholine. And there are several other things that can inhibit or stimulate the release of acetylcholine into the synapse. So what can stimulate the release of acetylcholine? Well, black widow spider toxin is one worth knowing. So with black widow spider toxin, the synapse is going to be flooded with acetylcholine. And that's going to cause a spastic paralysis. So black widow spider toxin can flood the synapse with acetylcholine. And then one toxin that can inhibit acetylcholine release is botulinum toxin. And this causes a flaccid paralysis. You just can't contract your muscles without acetylcholine that's stimulating the nicotinic receptors at your muscles, so you get flaccid paralysis. And then the diagram says 4 fates of acetylcholine. So you successfully released acetylcholine into the synaptic cleft, but what happens next? Well, one of 4 things can happen to that acetylcholine. First of all, it can do what it's intended to do, which is to bind to its postsynaptic receptor. Second, it can bind to an autoreceptor on the presynaptic cell, which regulates further acetylcholine release. Third, it can simply diffuse away from the synaptic cleft. And the fourth thing that can happen to acetylcholine, it can be degraded by acetylcholinesterase, which cleaves acetylcholine into choline and acetate. That choline can be recycled and transported back into the cell by the sodium cotransporter. And what drug category would inhibit this cholinesterase from degrading acetylcholine? The cholinesterase inhibitors, like neostigmine. Let's move on to the noradrenergic nerve terminal next. Now, in order to understand what's going on in the noradrenergic nerve terminal, we need to go over the steps of catecholamine synthesis. Now, it's pretty straightforward, but unfortunately you do need to memorize the steps and the enzymes and the cofactors, but not necessarily the chemical structures. So just listen for right now. We're going to write this out in a second. You start with the amino acid phenylalanine, and the enzyme phenylalanine hydroxylase adds a hydroxyl group to make tyrosine. Now, what happens if you're deficient in phenylalanine hydroxylase? Well, the phenylalanine builds up and leads to phenylketonuria, or PKU. So phenylalanine hydroxylase is an important enzyme to know. And this enzyme uses a cofactor called tetrahydrobiopterin, or BH4, which gets converted to BH2, dihydrobiopterin, in the process. And there's an enzyme called dihydrobiopterin reductase that converts BH2 back to BH4, so it's ready to go again. And if you're deficient in that enzyme, phenylalanine will build up and you'll get PKU. So there are two ways to get PKU. We'll talk more about PKU in biochem. So you've converted phenylalanine to tyrosine. Then the next enzyme is tyrosine hydroxylase, which adds another hydroxyl group to make DOPA. This also uses BH4. Now, DOPA stands for dihydroxyphenylalanine. So we started with phenylalanine, we added two hydroxy groups to get dihydroxyphenylalanine, or DOPA. The next enzyme is called DOPA decarboxylase, which removes a carboxyl group from DOPA, so you have dopamine. And there's an important cofactor for this enzyme, which is vitamin B6, pyridoxal phosphate. Then the enzyme dopamine beta hydroxylase comes along and adds another hydroxyl group to the side chain on dopamine, and that makes norepinephrine. And here, vitamin C is the cofactor. And then finally, the enzyme called PNMT, or phenylethanolamine N-methyltransferase, transfers a methyl group to the side chain, and that produces epinephrine. And this step is stimulated by cortisol. Now, in your study guide, there's a place for you to write down this pathway, so let's simplify it a little bit. You start by converting phenylalanine to tyrosine using the enzyme phenylalanine hydroxylase, and the cofactor required is BH4. Then you convert tyrosine to levodopa using the enzyme tyrosine hydroxylase and the same cofactor, BH4. Then you convert levodopa to dopamine using the enzyme DOPA decarboxylase and the cofactor vitamin B6. Then you convert dopamine to norepinephrine using the enzyme dopamine beta hydroxylase and vitamin C as a cofactor. And finally, you convert norepinephrine to epinephrine using the enzyme PNMT, and this is the step that's stimulated by cortisol. So those are the main steps of catecholamine synthesis. Now let's look at the noradrenergic nerve terminal, which is where norepinephrine is released. We start out by bringing tyrosine into the cell. And tyrosine comes into the cell using a sodium cotransporter, using the extracellular sodium gradient to bring tyrosine in. Once tyrosine's in the cell, the enzyme tyrosine hydroxylase converts tyrosine to DOPA, and there's a drug that can inhibit this step called metyrosine. Then DOPA is converted to dopamine by the enzyme DOPA decarboxylase. And we're not going to fill in all the enzymes on this diagram. Then dopamine is converted to norepinephrine, and norepinephrine is packaged in the vesicle. Then there's a drug that can inhibit this packaging of the norepinephrine into the vesicles, which is called Reserpine. And what substance causes vesicles to fuse with the cell membrane and release the contents of the vesicle into the synapse? It's calcium, just like in cholinergic nerve terminals. But besides calcium, what other substances stimulate norepinephrine release? Well, one thing is amphetamines. Some amphetamines will facilitate the release of norepinephrine into the synapse. And ephedrine works the same way. And then another substance worth knowing is tyramine. So the three substances I want you to know that can facilitate norepinephrine release into the synapse are amphetamine, ephedrine, and tyramine. Now, what can inhibit the release of norepinephrine? Well, the two drugs I want you to know are guanethidine and beryllium. Beryllium is a potassium channel blocker used as an antiarrhythmic for heart rhythm disorders. And one of the things it can do is inhibit the release of norepinephrine vesicles. Now, once the norepinephrine has been released, what can happen to it? What are the fates of norepinephrine? Well, it can bind to one of the adrenergic receptors on the postsynaptic cell, such as alpha-1 or beta-1 receptors, or it could bind to an alpha-2 autoreceptor on the presynaptic cell, which is going to inhibit further norepinephrine release. And there are other receptors on the presynaptic cell that can regulate this release of norepinephrine, such as the M2 muscarinic receptors. Think about it. If you have parasympathetic muscarinic activity going on, you want to simultaneously inhibit adrenergic activity. And then angiotensin receptors will facilitate norepinephrine release. So those are two of the fates of norepinephrine. It can bind to a postsynaptic receptor or it can bind to a presynaptic autoreceptor. A third potential fate of norepinephrine is reuptake, which you can see right here. One of the main ways that norepinephrine gets out of the synapse is it gets taken back up by the presynaptic cell. And there are several substances that inhibit this reuptake that are worth knowing. Cocaine, tricyclic antidepressants, and also the SNRIs, serotonin norepinephrine reuptake inhibitors, like venlafaxine or duloxetine. So cocaine, TCAs, and SNRIs all inhibit norepinephrine reuptake. So that's why cocaine makes the heart race and raises the blood pressure. That's why TCAs and SNRIs are useful in treating depression. The fact that ephedrine increases norepinephrine release explains why ephedrine supplements and energy drinks and appetite suppressants can be so dangerous. And this is why tyramine can cause hypertensive crisis in patients that are on certain medication. It all ties into norepinephrine release and reuptake. There's also a fourth potential fate of norepinephrine, which is that it can be metabolized, not necessarily right here within the synapse, but there are a couple of enzymes that do metabolize norepinephrine. One of those enzymes is catechol-O-methyltransferase, or COMT. Now the thing I want you to know about this is that it methylates norepinephrine. It's a methyltransferase. It transfers a methyl group onto norepinephrine. Another enzyme that can metabolize norepinephrine is monoamine oxidase, or MAO. This oxidizes norepinephrine. So the methyltransferase is going to methylate norepinephrine, and monoamine oxidase is going to oxidize norepinephrine. Now, both of these reactions are going to make norepinephrine unusable as a neurotransmitter. So if you have a pheochromocytoma that's making lots of catecholamines like dopamine and norepinephrine and epinephrine, these two enzymes, COMT and MAO, will convert those catecholamines to things like metanephrine and normetanephrine and VMA, which you can test for in the urine to help make the diagnosis. Specifically, these enzymes convert dopamine into HVA, or homovanillic acid. They convert norepinephrine into VMA, or vanillylmalonic acid, and they convert epinephrine into metanephrine. Now, you're going to see this again when we cover pheochromocytoma in the endocrine section. And you'll also see COMT and MAO when we discuss Parkinson's disease, because Parkinson's disease is characterized by a deficiency of dopamine in the brain, and we use drugs that inhibit COMT and inhibit MAO as a way to raise dopamine levels in Parkinson's patients. And MAO inhibitors, or MAOIs, have also been used as antidepressants. Now, we don't really use them much anymore, but increasing norepinephrine can treat depression, just like we saw with the TCAs and the SNRIs. So again, it's important to understand both the catecholamine synthesis pathway and also the drugs that affect adrenergic neurotransmission. All right, before we get to the end of session quiz, let's do a quick review. In this whiteboard review, I've listed several drugs and toxins on the left, and we're going to match them up with their actions on the autonomic nerve terminals. The first one here is amitriptyline, which is a tricyclic antidepressant, a TCA. And we said that those inhibit norepinephrine reuptake. The next one is amphetamine, which we said stimulates norepinephrine release. Then we have black widow spider toxin, which stimulates acetylcholine release. Botulinum toxin inhibits acetylcholine release. Beryllium inhibits norepinephrine release. Cocaine inhibits norepinephrine reuptake. Guanethidine inhibits norepinephrine release again. Hemicolinium inhibits choline transport. You can remember the choline and hemicolinium. It's pretty obvious. Reserpine inhibits the packaging of norepinephrine into vesicles. And visamicol, again, the col tells you it inhibits packaging of acetylcholine into vesicles. It's all right. You did a great job on that one. And for your reward, since we mentioned amphetamine, I'm going to draw you a little Heisenberg. Now it's time for the end of session quiz. So go ahead and work through the questions, and then we'll go over these answers together. All right, first question. Which three enzymes are required to convert phenylalanine to dopamine? So you need phenylalanine hydroxylase, which converts phenylalanine to tyrosine. You need tyrosine hydroxylase, which converts tyrosine to dopa. And you need dopa decarboxylase to convert dopa to dopamine. Next, a patient develops paralysis after being bitten by a black widow spider. Is this a spastic or a flaccid paralysis? And what other autonomic symptoms might be seen? So remember, black widow spider toxin stimulates the release of acetylcholine, which is the neurotransmitter at the neuromuscular junction. So that causes muscle spasms and possibly some spastic paralysis. And the other symptoms might include salivation, diaphoresis, lacrimation, all from excess acetylcholine. Everything gets leaky. Next, what substances inhibit the reuptake of norepinephrine? We mentioned cocaine, tricyclic antidepressants, and SNRIs. Next, what substances stimulate the release of norepinephrine from neurons? So calcium is the normal trigger for the release of norepinephrine. And then also we mentioned amphetamines, ephedrine, and tyramine. All right, I'll see you next time.